day everyone! Today, let's talk about what happened during mitosis. But first, what is mitosis? Mitosis is a type of cell division that results in two daughter cells having the same number and kind of chromosomes as the parent nucleus. Before we start, we must remember the acronym IPMAT for the process during mitosis. IPMAT stands for interphase, prophase, metaphase, anaphase, telophase, and cytokinesis. Interphase, the chromosomes duplicate and become two identical chromatids, joined at the centromere. In prophase, the chromosomes condense, and in the cytoplasm, spindle fibers form. Metaphase, the nuclear membrane breaks apart, the spindle fibers catch the chromosomes, and the chromosomes line up with the equator of a cell. In anaphase, the spindle fiber shorten and the centromere divides so that each chromosome becomes two separate chromatids. In telophase, the nuclear membrane forms from each seed of chromosomes. Chromosomes break back out in a new nucleus and the spindle fibers break down. In cytokinesis, the cell membrane pinches into separate six of chromatids into two identical daughter cells with the same number of chromosomes. In real diagram, this is what it looks like.
Meiosis, special type of cell division where the sex cells are created. It involves two rounds of division, the meiosis 1 and the meiosis 2.
During prophase 1, the complex of DNA and protein known as chromatin condenses to form chromosomes. A large structure called the meiotic spindle also forms from long proteins called microtubules on each side of the cell. In prophase 1, nuclear envelope disappears, tetrads are formed, and crossing over takes place. In metaphase 1, the nuclear membrane is completely disintegrated. Spindle fibers are now completely attached to the tetrads, and tetrads move to the metaphase plate.
Anaphase 1 is the third stage of meiosis 1 and follow prophase 1 and metaphase 1. This stage is characterized by the movement of chromosomes to both poles of meiotic cell via microtubules network known as the spindle apparatus. This mechanism separates homologous chromosomes into two separate groups, and telophase 1 is the phase when the chromosomes are finished moving to opposite ends of the cell. This will be followed by cytokinesis producing two daughter cells. Thus, after meiosis 1, each cell will have haploid set of chromosomes, but each chromosome will still be comprised of two sister chromatids.
Cytokinesis comes after telophase 1. The cytoplasm divides into two new daughter cells.
Prophase 2 is the phase that follows after meiosis 1 or after interkinesis. If present, if interkinesis takes place, the nuclear envelope and the nucleus disintegrate during prophase 2. The chromosomes are condensed, the centrosomes replicate and move towards the opposite pole. Metaphase 2 is the second stage in meiosis 2. The cell is in metaphase 2 when the chromosomes align themselves along the metaphase plate through the facilitation of the spindle fibers. The spindle fibers are now attached to the two kinetochores contained in the centromere of each chromosome.
Anaphase 2. Sister chromatids of each chromosome separate and move towards the opposite poles. Telophase 2. Nuclear membrane forms around each set of chromosomes, and cytokinesis occurs.
Cytokinesis, the final stage where the cytoplasm divides into 4 haploid daughter cells.